Maybe, I don't know. I think
One more thing before we get started. This is the view that online are going to see. Basically stay between the green.
yeah.
I'm just stare. And also because we are having it's possible that Lex will say, hey, there's a technical problem.
Again, it's not very good. Yeah. Okay.
If we have a problem, we can try this one. Or we can, worst case scenario, we find it in color.
Okay. I'm fine talking on this too. Is this on?
Yeah, it's not. Should we get started? You're up.
That was such a shit show.
Okay. I can't even try. I can't even say your name as fast as
This one, it's like a joke of a class though. It makes me smile every. It's like my grades so many bad professors and science are just like writing something really. What are that's it's for healthcare ops. It's actually in this class. Really? I can't find it.
Okay, welcome everyone. Welcome everyone to our BME seminar series hosted by the Department of Biomedical Engineering at Columbia University. My name is Henry Hess, and I'm a professor of biomedical engineering here at Columbia. Today is our final BME seminar of the semester. We continue to offer this lecture in a hybrid format with both virtual and live audience. Our speaker will present for approximately 40 to 45 minutes, and we will leave 15 minutes at the end for questions and answers. For our virtual audience throughout the presentation, please use the Q&A feature at the bottom of your screen, not the chat, to submit your questions. We'll try to answer as many questions as possible during the Q&A session at the end. For our live audience, you will have the chance to ask your questions during the Q&A. Today, we are excited to welcome Professor Aaron Streets. Dr. Streets received the Bachelor of Science in Physics and the Bachelor of Arts in Art at UCLA. He completed his PhD in Applied Physics at Stanford with Dr. Stephen Quake, and Aaron then went to PhD in China. He was an international postdoc fellow and a fourth postdoc fellow, and worked with Dr. Yan-Li Wang in the Biodynamic Optical Imaging Center, BIOPIC, at Peking University. Aaron joined the faculty of UC Berkeley as an assistant professor in bioengineering in 2016 and is currently a core member of the biophysics program in the Center for Computational Biology, and he is a Chan Zuckerberg BioHub investigator. Aaron has received the NSF Early Career Award and was named the Pew Biomedical Scholar. Welcome, Professor Streets.
Thank you. Thank you for the invitation. It's fantastic to be here. It's fantastic to be anywhere. Really. Great. As Henry mentioned, I'm in the computational biology program, bioengineering, and biophysics. And so my lab has students from all three of these programs, which works really well because we sort of focus on building new tools, both hardware and software tools for studying single cells. And so I'm going to try to tell you two stories today. One is about a technology that's fairly mature enough so for us to be able to start to learn something potentially interesting about biology. And then the second story is about a new tool that we've been working on for using long-read sequencing technologies like Nanopore. So to start, how do we define the identity of a cell? How do we quantify that identity? And the answer typically is by detecting the abundance and the composition of a set of molecules. And those molecules can lay in any one of these layers that I've depicted here or beyond. And many of these molecules can be detected very accurately by encoding their identity into an optical signal and then using a microscope to detect that signal. And the microscope, of course, is the first way that we've ever detected the identity of single cells. It's the first way that we knew that there were cells. And it still remains one of the most powerful ways of quantifying cellular identity, because with optical amplification, you can get down to the sensitivity of being able to detect a single protein or a single RNA transcript. However, the challenge with microscopy is that you have limited bandwidth. There's only so much space in the visible spectrum or in the UV and IR spectrum. How are we doing on sound?
Alexis tells me it's good all the time.
It's good all the time. Okay, so this little thing that we're seeing is not.
Yeah, which is confusing me, but just go with it.
Perfect. Yeah. We can only fit so many fluorophores in the visible spectrum. So we can at best maybe see 10, 20 different molecules if you have a really high quality spectral flow cytometer. But for some of these layers of information, we have 3 billion letters in our genome. We have tens of thousands of genes, hundreds of thousands of proteins in proteoforms. And so when detecting the identity of a cell by quantifying molecules on the genome-wide scale, DNA sequencing is a really powerful tool. If we can encode the identity of these molecules into DNA, then the DNA sequencer provides a really highly multiplexed microscope to read out millions of different molecules at the same time. The key in that case, you need to somehow turn the identity of those molecules into DNA, which you can't do for all of these molecules. Techniques like single-cell RNA sequencing nowadays have been really powerful for getting lots of information about gene expression from lots of cells and giving us the ability to use statistical algorithms to categorize cells by their gene expression profile. Of course, the challenge with sequencing is that you destroy all spatial information. You lose the position of origin of those molecules. And so in my lab, we like to think about how we could combine these two different kinds of measurements using microfluidic tools to allow us to couple measurements of optical signal with genomic measurements. And when I say genomic measurements, I basically mean turning the proteome or the transcriptome into DNA so that you can use the DNA sequencer to read it out. And doing this at the single cell level. And really nowadays, measuring the transcriptome or the genome of a single cell is not hard. We have good molecular tools for making that measurement. The hard part is doing it on the single cell level and doing it for many cells. And so if you look at, this is a plot from a review that we wrote. If you look at the number, this is for single cell RNA sequencing, where we're looking at the gene expression profile using sequencing of many cells. And if you just look at the number of cells that any given publication was able to analyze in that study, it sort of increases exponentially, and there's this inflection point right around 2014, where it goes from tens and hundreds of cells to thousands. And this is really because of the introduction of microfluidic tools for helping increase the throughput of single-cell RNA-seq, catching cells, putting them into wells really quickly, and allowing us to do amplification and DNA sequencing. And so over the past 10 years, a lot of the development of single-cell genomic analysis has been in this sort of space, increasing the throughput, of how many cells we can measure per experiment, and increasing the sensitivity, how many genes we can measure per cell. One of the things that we've been interested in the past couple of years in my lab is trying to develop new tools, both hardware, molecular, and software tools to expand in this new dimension. How many different kinds of molecules or different layers of information can we measure in the same cell at the same time for many different cells? Can we measure the proteome? Can we probe epigenetic state while also looking at gene expression? And then once we develop a tool where we can analyze new kinds of multi-omic data, multimodal data for single cells, then the question becomes, how do you analyze that data? Now you have thousands of cells with many different dimensions of identity, different facets of the cell's molecular identity. And you have enough variables where it's hard to visualize those on a low-dimensional space. And so how do we combine the information the right way, not only so that we can get a holistic interpretation of what a cell is, but also so that we can understand the relationship between these layers. How does gene expression relate to epigenetic state? How does protein abundance relate to gene expression? So the two stories I'm going to tell you, one is about using a fairly mature multi-omic technique for studying T cells, and then the other one's going to be a new approach for looking at different kinds of epigenetic states on single cells and single molecules. The first story, it has been a really fruitful collaboration with Nir Yosef and Ellen Roby. Nir is a professor of computational biology at UC Berkeley, and Ellen's an immunologist. We had Zoe and Adam, who were fantastic graduate students, leading this project. And the goal of this collaboration was to use single cell genomic analysis to understand how we develop CD4 and CD8 T cells in the right amount. So T cells, our immune cells, are developed, are produced from our thymus, this organ just behind our sternum. And we need a lot of killer T cells, helper T cells to help our body respond to infection. And so the way these cells are produced is hematopoietic precursors migrate from the bone marrow into the thymus, where they get positively selected, and they'll express these two proteins on their surface, CD4 and CD8, co-receptor. And so these progenitor cells we refer to as CD4, CD8 double positive, because they have both of the proteins on their surface. And what I'm going to do is, many of you might already know this stuff, but I'm just going to go through it anyway, just in case we're all on the same page, because usually in bioengineering departments we have a lot of different. So if I'm saying something you already know, just nod along. And so we call these double positive T cells. They have both CD4 and CD8, and these are bipotent. They can differentiate into CD4 or CD8, helper or killer T cells. And they undergo this differentiation as they migrate through the thymus, picking up signals from the stromal cells and the antigens that are being presented, signaling to the T cell receptor. And then that T cell receptor signaling and the specificity of the T cell receptor to different antigens in the thymus end up dictating the fate of these cells in a way that we know a lot about, but that we still don't fully understand what all the transcriptional and the epigenetic regulatory events that are upstream of this branching point. How do we, if we look at a double positive T cell, we don't know what it's going to be. But if we look downstream far enough, we can start to see the difference between some of the transcription factors that are master regulators for one or the other branch. But what controls that decision? It's both a very classical system for studying cell fate, and it's a really important process in our body. So we were interested in studying this process in the mouse model. And in part because Ellen has been doing this for decades, and she has defined, and she and her colleagues have defined a lot of the transcriptional and proteomic events that dictate this lineage. So what I'm showing you here is some of the intermediate states between the double positive population and single positive population. And these discrete states are defined by sets of surface markers, right? So you use flow cytometry. We can label in CD classification and fluorescent antibody, we can label a certain panel of markers that define these states. And by the presence or the absence of these markers, we can define some of the intermediate states, mature double positive or immature single positive. We know kind of more or less the timeline. But, and if we were to do this, let's say we're looking at CD4 and CD8, this is a FACS plot or flow plot, where we're just plotting the abundance of CD4 and CD8 protein. And you can see the double positive state, both proteins. You can see the single positive states, and you see the states in between. And this is sort of a really coarse way of defining this timeline, but of course we can use many other proteins to define it with higher resolution. What we wanted to do is we wanted to approach this problem with single cell analysis so that we could fill in the continuum of states in between these discrete states and ask what's happening just here, right before this, the last state that we know is bipotent and the first state that we know is already committed, what's happening at that branch. And so, as I mentioned, the way we typically analyze this system and sort of most of the information we have in the literature is by labeling proteins with antibodies that have fluorophore and quantifying the protein abundance with flow. The technology that we used for this experiment is a technology called CiteSeq that was developed at the New York Genome Center by Raul Satija and Peter Schmeiberg. And what CiteSeq does is, instead of using fluorophores to label the antibodies, it puts a DNA barcode that's unique to the antibody on each antibody, and therefore you can use sequencing to read out the identity of that antibody to the fluorescence, which gives you access to a much wider bandwidth. So now you can look at hundreds or thousands of proteins at the same time. And the trick with this technology is that in addition to this unique barcode, there's a poly A sequence at the end of this barcode, which means that if you do traditional single-cell RNA sequencing, which typically captures the polyadenylated tail of the mRNA, you can get both gene expression and surface protein abundance in the same experiment. So the way that this experiment works is you have a panel of antibodies with unique barcodes. You stain your cells with this panel. You wash away all the antibodies that don't bind. Then you put your cells into a microfluidic device that puts cells and barcoded beads into droplets. This is a commercial technology provided by 10x Genomics, but it puts beads and cells into droplets and allows you to barcode the transcriptome and the proteome of each cell so that when you sequence it in a sequencer, you get back this really rich data set. The data set is this matrix where you have a bunch of cells, thousands of cells, and for every cell you have a measurement of its gene expression by a variable for every gene in the genome. And you have a measurement for the abundance of the proteins that you analyze with your panel. And this is a huge data set. And you can imagine that we can't use traditional two-dimensional plots to analyze the identity of cells, right? Because there's so many axes that it would be impossible to visualize. And so the first question was, what's the right way to analyze this data? And so this is, and not only how do we process it, but how do we interpret it? And there's a lot of important details about the way that these different measurements are made. And so we developed a model that's based on a variational autoencoder with Professor Yosef and a joint student that we call total VI. And what it is, it assumes a couple things about the data. So our goal is to combine the measurement of all the genes and all the proteins into a low-dimensional representation for each cell, right? So that we can have a better idea of where that cell sort of fits in state space. And this model makes a couple assumptions. The first assumption that it makes is that the data is very sparse. In other words, you don't need 10,000 by 200 variables to assess most of the information in the system. Many cells are similar to each other. Many genes are expressed in a sort of co-regulated fashion. Many proteins do or don't have new information. So the idea is you can, in theory, compress this down to fewer dimensions and still contain the vast majority of the information. That's one assumption. The other assumption is that the RNA counts, the measurement of gene expression for each cell, for each gene, follows a negative binomial distribution. So if you make this assumption, instead of having an absolute count for every transcript, you can sort of model a probabilistic distribution that cell is expressing that gene with this mean expression level and this confidence. So you can kind of develop some sort of statistical test about the expression level for every gene in every cell. And we also assume that the protein measurements follow a negative binomial mixture model. We assume that there's a background. So these are two very different measurements. One's sparse, one's noisy. Both noisy. And so by making these assumptions and putting this through this variational autoencoder, this model, it learns at every iteration, it learns a 20-dimensional matrix that will define all the information for every cell. And at every iteration of the model, it reevaluates the likelihood that a certain set of parameters is estimating the value for that gene or for that protein, and it updates that estimation based on all the data, the expression of all the genes and all the cells and all the proteins. So in this way, we can learn this low-dimensional latent space. And if we label cells also by the experimental batch or other sorts of, which mouse did it came from, we can effectively remove or control for categorical and continuous covariates that might be uninteresting variation in the data and focus just on the differences in cell states. So that was the computational tool that we used to analyze the data. And then we also used a couple biological tools. So in addition to, one of the great things about studying this system in the mouse is that if you sample a thymus from a 3-week-old mouse, these T cells are constantly differentiating. And so any thymus has all of the states from double positive to single positive. But those double positive cells, we still don't know their fate. So in addition to the wild type mouse, we also use these lineage restriction mice. These are TCR transgenics and MHC knockout mice that only produce CD4 or CD8 single positive cells. So they have the double positive progenitor, but those cells, we know what their fate's going to be. And so this way, we can get a little bit of a way to stratify our data, so we can have these double positive cells that look like the ones from the wild type, but we know a little bit about their future of their lineage. And so we ran an experiment. We had about 7 different genotypes, all the transgenics and the knockouts. In total, we sequenced 80,000 cells. We had about 20,000 genes per cell and 100 proteins. And we put this data through the total VI model and just look at the 2 principal components, or in this case, a UMAP dimensionality reduction of the 20-dimensional latent space. We can see all of our cells in this one 2-dimensional space. And so each dot is a cell, and its position on this 2-dimensional axis is a representation of the 20-dimensional state, which is a representation of the whole data. And the blue cells are the lineage restricted mice that only make CD8. The red cells are the lineage restricted mice that only make CD4, and the purple cells are the wild type. And you can see that we sort of see this bifurcation, and we can ask questions about gene expression and protein abundance for every cell. And we can verify which branch is which. So the double positive cells are over here on the left, where you see, and here I'm plotting the color is proportional to the number of transcripts for CD4 and CD8, or the number of proteins for CD4 and CD8. And so you can see the double positive cells have both CD8 and CD4. And the single positive cells either have CD8, both protein and transcript, or CD4 protein and transcript. So this is, we think this is spanning the space. And all these in between, all these cells in between are somewhere, on this trajectory. And so the next thing we can do is we can use clustering to kind of define a minimum spanning tree. This is an algorithm developed by Kelly Street, a graduate student in Sandrine Dubois' lab at Berkeley, called Slingshot, and it maps every cell to this linear trajectory through this state space and gives that cell a score. And that score is, kind of a way to assess where that cell is along this trajectory, either on the CD4 or the CD8 branch. And so now with that number for every cell, we can collapse all of the gene expression in the surface protein abundance onto a pseudo-temporal timeline, where the time axis is not real time, but it's sort of how far along the progression a cell is. And so now what I'm doing is every row is a gene, and every column is the average expression across all the cells at that state, right? And so you can see, like, these are genes that light up early in a double positive stage, and these are genes that light up later in a single positive stage. These are proteins. And we know that trajectory for each lineage because we have the lineage restricted mice. And the key point was that with the total VI, we could remove the batch effects that would otherwise be associated with different mice, because the different genotype mice have different gene expression. If you were just to plot these naively onto a linearly deconvolved reduced dimension, you'd see that the cells would cluster by which mouse they came from. We can sort of remove that variation with total VI and just look at it so that the double positive cells over here overlap. And so this, we think, or we propose, is also a measurement of what's happening in the wild type system. So now we can use that surface protein data to make these sort of flow plots. Right, so this on the left was what I showed you before, where you have fluorescence to measure the abundance of CD4 and CD8 in the double positive and single positive. This is the data that we got from sequencing the antibodies for CD4 and CD8. So you can see that you get the double positive population, every single positive population. But now for every cell, we have its pseudotime point. And so now we know, where that given cell was in its trajectory. And we can start to ask questions about what's happening as these cells bifurcate and go into the single positive state. And we can do that for each mouse. And so for the wild type, you see the branching in both directions. You see the CD4 cells losing its surface expression of their CD8 in the CD4 lineage. In the CD8 lineage, you see this other interesting phenomenon where the cells, and this had been observed before with flow, but the cells start to lose CD8 surface abundance and then come back through this double positive phenotype before they become mature CD8 cells. And you can see this because we have the color bar, which tells you where the cells are in pseudotime. But you can imagine how this would be hard to evaluate just with the surface protein abundance because, they look kind of like the double positive cells. It's just that they happen to be a little bit more mature and already committed to CD8. And so we can break down this whole trajectory in this flow, this sort of in silico flow data And we can ask, of all the 100 proteins that we profiled, what is the right combination of surface proteins to give us a higher resolution to be able to pull out some of these intermediate states? We can define a new set of surface proteins that more precisely pull out these pseudotime points because we have the pseudotime information. And so that allows us to now identify cells in this trajectory just by their surface protein. So we can pull out, these double positive, these sort of intermediate states. We can pull out the semi-mature, single positive cells and the mature. I mean, some of this you could do already, and we added a little bit of resolution in pseudotime. So now let's get at the question that we started, which is what drives this branching? And this is a hard question, and I'll show you how we started to think about it. It's not the end of the story, it's like the beginning. But when asking what happens upstream of branching, we have the data, and we know the fate of these lineage restricted mice. And so what we can do is we can ask a question. We can do a statistical test. What genes are being differentially expressed between double positive cells from the two lineages that are just upstream of the branching point? And we can ask, we can even ask more potentially, what's the earliest time we see differences in gene expression between these two lineages? And so, here what I'm plotting is differential expression between certain markers as a function of pseudotime. And you can see that the CD4 and the CD8 transcript of protein start to diverge in the way that you expect. If you look at the gene expression, you see that the transcription factors which regulate commitment to either one of these lineages, these are the 3 transcription factors that are known to be master regulators of this commitment. You can see them diverge a little bit earlier than the markers do. And then we also found a couple genes that are known to be part of the T cell receptor signaling pathway also start to diverge. First, they get upregulated in the CD4, and then later they get upregulated in the CD8. And so we were interested in what's happening earliest. Like what's the first time we can see a difference? And so what Zoe did, she came up with this really beautiful question, a test. And she said, okay, so here are all the genes that are differentially expressed as early as possible. To go one step earlier, she asked, if we have a list of transcription factors with known targets database, in this list of differentially expressed genes between the two lineages, which transcription factors, the targets of which transcription factors are enriched in this list? What transcription factors are most likely governing this differential expression? And in this way, she was able to make a little bit of a hypothesis about which transcription factors might be good targets for perturbing commitment to one of the things. And one of the things that she found is that the transcription factors that are the targets of 3 branches in the T cell receptor signaling cascade were all enriched in one or the other pathway. So these are 3 branches of TCR signaling. We have this calcineurin NFAT branch, and then this NF-kappa B branch. And these are the transcription factors at the end of that signaling cascade. And we saw that the calcineurin NFAT was sort of exclusively regulated in early commitment to CD4. And so Zoe found that there's a small molecule, cyclosporin A, that will block calcium signaling to calcineurin and effectively block activation of NFAT. And so she said, let's see if we can use this drug to inhibit commitment to CD4. How are we doing? So this was a really beautiful experiment that Ellen's lab came up with, where Laura extracted neonatal thymic slices and put them in a culture to keep them alive so the T cells could differentiate. And then we challenged those thymic slices with this drug. And then instead of doing RNA-seq, we could use that panel of surface antibodies that we had previously defined to identify different substates, to just use flow to ask what's the abundance of these different states after we challenge this slice with the drug. And the long and short of it is that what we found is that when we challenged these thymic slices with the drug, we decreased the number of cells in that semi-mature CD4 state without perturbing the number of cells in the CD8 lineage. And interestingly, we weren't able to block commitment to these mature cells. And the hypothesis that we have is that some of these cells that were mature by the day 3 when we analyzed the flow had already committed to CD4. So the drug didn't inhibit that early signaling, but these cells in the middle that had already committed to CD4 but weren't quite mature yet, those are the ones that ended up getting inhibited by the cyclosporine. So this is a kind of really nice example of how we could generate a hypothesis using this large data set and go and test a hypothesis in vitro or in situ kind of model. And there's a lot more in this paper that Laura wrote, including this sort of this proposed model. In addition to these particular transcription factors, we're able to see 2 waves of TCR signaling. And so the model that Ellen is proposing with this data is that instead of this fork in the road sort of lineage commitment where the cells either choose to go CD4 or CD8, all of the cells audition for the CD4 fate by a wave of TCR signaling and testing antigen specificity as it migrates to the thymus. And cells that win that audition go and become CD4, and cells that lose that audition then go down the CD8 path, but then they need to be specified by a second wave of TCR signaling, and they can either die or become CD8. So instead of a fork in the road, it's more like a freeway exit, that you can take. And we're still exploring some of the nuances of this model with follow-up experiments. So this is an example of using measurements of molecules like gene expression and surface protein to identify cell states, to identify what's happening. But you can see that what we were trying to get at with this information is why is it happening? What's sort of epigenetic regulators that are driving this fate, which is defined by the abundance of transcripts and surface proteins. And so if you wanted to move upstream of the dogma, the next question is, well, how is the genome being regulated? What signals are regulating the genome to give rise to this gene expression? And to address that kind of question, we need to look inside the nucleus. And we know that proteins in the nucleus are the primary drivers of regulating, repairing, modifying the genome. And so typically when we want to probe the epigenetic state of a cell, what we do is we ask where in the genome is DNA modified or histones modified or is a protein interacting with DNA? Where is a transcription factor binding? Where are RNA polymerases? Where is a histone modified? And so by asking these kind of questions, a typical suite of tools that we use are genome-wide measurements like ChIP-seq or cut and tag, which ask that question: where in the genome, in the full genome, is a protein or a histone of interest located? And the way that most of these techniques work, whether it's ChIP-seq or DAM-ID or cut and run, is that they use an enrichment strategy. Right? So the idea is you target the protein that you want, and you use one or another of techniques. You can do immunoprecipitation, or you can do TN5 tagging. But whatever you do, you enrich for fragments of DNA that are bound to the protein of interest or the histone of interest. You enrich your DNA for those fragments, and then you sequence those fragments using the DNA sequencer. And what you're looking for here is not just sequence, but you're looking for where the reads map, and the abundance of reads at a given locus is going to be proportional to the likelihood that your protein of interest was down there, was targeted, right? So this is a coverage-based proxy for protein interactions. The challenge with this technique, there's a couple of times, it's ensemble measurement. First of all, these techniques are incredibly powerful. It's the way that we've mapped all the regulatory elements of the genome and code. We have really comprehensive maps of transcription binding landscape, factor binding landscape, chromatin modifications using these techniques. But there's some gaps in these techniques. And one gap, literally, is that these short sequencing reads, 200 base pair sequencing reads that we get off of the Illumina sequencer, it's hard to confidently map them to regions of the genome that are highly repetitive. Right, so there's areas in the genome like the centromere, which are these long stretches of tandemly repeated 170 base pair monomers that are repeated like for millions of base pairs. And so if you get a 200 base pair sequencing read and you try to align it to the genome for reads that come from the centromere, for example, we can't align them. It's really hard. Usually we throw those out when we do the assembly. Also, because of these are ensemble techniques, it's really hard to ask. Let's say that you have, two peaks, and you want to know, is it that half the cells the protein's binding at this peak, or half the cells the protein's binding at that peak? or is it half the cells are binding at both peaks and half the cells are binding at no peaks? It's hard to answer that question about distribution with these ensemble measurements, right? And so in another really awesome collaboration with Aaron Strait, you can tell that I only collaborate with people that have similar names. Aaron Strait is a professor at Stanford, and Norma is the director of sequencing at BioHub. And we had two grad students in my lab, Nick and Annie, who were phenomenal graduate students. Nick graduated last year. And then Kausic and Owen from Aaron's lab. This was a project that we worked together to come up with a new way of mapping protein-DNA interactions using long-read sequencing. And we call this technique DMelo-seq for directed methylation and long-read sequencing. So there's a couple potential benefits of this technique, and then I'm going to tell you the benefits, and I'm going to try to convince you that it does those things. So the first one is that if we can map to nanopore sequencing and packed biosequencing, instead of giving us short 200 base pair sequencing reads, we can get 1,000, 10,000, 100,000 base pair long reads, which give us the ability to align to places like the centromere, which are highly repetitive. So in theory, we can map protein-DNA interactions into repetitive regions. Also, if we have really long reads that span 2 binding sites, we can ask on the same molecule, are both proteins bound or is one bound or the other? And one important point about these long read sequencers is that in Illumina sequencing, typically what you do is you enrich, like I said, you amplify your DNA. And so every read that you sequence is an amplicon. It came from some template from PCR. Here, we're not doing any amplification. So every read that we sequence came from a molecule that came from a cell. And so these are native chromatin molecules. And so we can map, binding onto the original molecule. Also, these long read sequencing tools, they're capable of reading DNA modifications as well. They're sensitive to methylation, for example, of DNA. And so in theory, you also get back simultaneous endogenous CpG methylation on that same molecule. So you can look at the relationship between DNA methylation and protein binding. Because every molecule came from a cell, in theory, the number of reads that we get, the binding frequency of a given protein should be proportional to the number of cells that had that event. So it's a single molecule / pseudo single cell technique. And then also because these reads, there are some heterozygous sites that are unique to the allele from which that molecule came. And because we have long reads, it might be able to span many of these heterozygous sites. That means we can phase reads, which means we can map reads back to the maternal allele and profile haplotype-specific protein DNA bindings. This is the promise of long-read sequencing for DNA protein interactions. So this is the way the technique works. So we take a bunch of cells, we extract the nucleus, and permeabilize it, and we target a protein. Let's say this is our target protein. And we find an antibody that's really good against that protein, and we permeabilize the nucleus. This is like TIP-seq or cut-tech. And we stain the nucleus with the antibody, and we wash away all the unbound antibodies. And then this is the secret sauce. We have this molecule that's a complex between protein A, which will bind to the antibody from the same species, and then this DNA methyltransferase. This is a molecule which will methylate any adenine that it comes near, any A that it comes nearby. So anywhere that your protein goes, if we add a methyl donor, if we activate this methyltransferase, then we will effectively mark the DNA nearby the protein binding position with a methyl adenine. And the important thing about this is we have a lot of cytosine methylation in our genome. Humans don't have adenine methylation. So this provides a mark that's distinct from anything that you would see in the genome already. So it's like a different color. So then you extract DNA from the nucleus and you try to maintain these long fragments of DNA. And then you put these molecules of DNA onto a nanopore sequencer. This is a USB device. It's about the size of this, this pointer. And we have a couple of them in the lab. And overnight, or over the course of maybe a day and a half, you can sequence these long reads. And the way that it works is these long molecules are pulled through these nanopores, and there's a current across the pore. And so if you measure that current, as different bases go through the pore, it has different impedance. And bases that are modified have even a different impedance. And so you can make a current measurement, an optical measurement, to measure out the base and the modification of that base. So this is how nanopore sequencing works. And it's also sensitive to both adenine methylation, but also to cytosine methylation. And so now if you have one of these long tandemly repeated stretches in the genome, we have these long reads that we can align to that area of the genome. And then anywhere that we see methyladenine, and if we calculate in bins the ratio of methyladenine to just non-methyladenine, the peaks in that signal should correspond to places where a protein was likely bound, right? And then also we have cytosine methylation that also will indicate where the DNA was endogenously methylated. So this is what happens when we target CTCF. CTCF is a molecule that's found all over the genome. It mediates loops between topologically associated domains, it's a repressor, it's a binding factor. And it has, the reason why we chose CTCF, it has a very narrow footprint, has a very well-defined binding motif, meaning there's specific sequences that we know it should bind in the genome. So there's a reference, round two. It has an extremely well annotated binding profile using ChIP-seq encode. So we have places that we can look for positive control. And what I'm showing you here on the right is each row is a single molecule. And the color is how many methyladenines there were in that bin. And we're ordering the molecules by places that have the strongest ChIP-seq signal in encode. So the top, quarter of these sites have the strongest ChIP-seq signal, and the bottom quarter of these sites have the weakest ChIP-seq signal. And then in this figure, we're averaging the signal across all of these quartiles, and we're plotting the proportion of methyladenine as a function of bins. And so, the peak is associated with where the CTCF is binding. And there's a couple of interesting things about this profile. So first, you see that there's a really strong dip, and we're aligning all of these reads to the middle of the CTCF binding motif. We know where the middle should be, right? And so this dip, so we call the resolution of this measurement like the width of this peak, but this dip in the middle is the place where the CTCF molecule is actually binding to the DNA and it's blocking methylation. So that it's like a shadow. So in fact, that's where the CTCF molecule is found. What else is interesting about this figure is that you see these sort of ripples that look like, if you're a microscopist, it looks like a diffraction pattern when you're looking at a single molecule, these interference. It's not an interference pattern per se, although I like that analogy. These are nucleosomes on either side of the binding motif that are blocking methylation. It's harder to methylate DNA inside the nucleosome than in the linker DNA. So we're sort of looking at accessibility as a background signal on top of the signal. And what's great about this data is, like I said, we also have CpG methylation on the same molecule because nanopore sequencing can read that information out. So you can see it. So in red now, I'm showing you the concentration of methylcytosine. And you can see this really nice anti-correlation between CpG methylation, which is known to block CTCF binding, and the binding. These are average signals. These are the single molecules. So we can do joint methylation and binding, and some of these reads span 2 CTCF binding sites on the same molecule. So these are a bunch of sites these are a bunch of reads that span adjacent binding sites. They're not all the same distance apart. So we use this buffer in the middle, and we sort of anchored the middle of each side to the motif. And this is an example of what I was saying. You can see for all of these adjacent motifs, you can see some cells that had CTCF binding on one side, some cells that had CTCF binding on the other, and some that had both. But also, out here with some that have none. And so, in theory, you can start to assess coordinated binding of adjacent sites. And one of the things that's really nice about this approach is that now we can look at how this coordinated binding occurs on phased reads. So this is an example of an imprinted genomic region that contains the IGF2 and ACE19 locus. These are 2 genes. And genomic imprinting is a phenomenon where you see allele-specific gene expression. So you see expression of one gene coming from one allele or another gene from another allele. And this is a classic locus where you have sort of epigenetic regulation of one of the genes on either allele. So in this case, on the maternal allele, CTCF binds and represses expression of IGF2 and activates expression of H19. Whereas on the paternal allele, you have CpG methylation at this control region in the middle, and you have activation of IGF2 and repression of H19. And so if we take all of our reads and align to this control region in the middle, we can figure out which allele it came from because they span these heterozygous sites, which allows us to phase the reads, allows us to identify. These are sites that are different DNA variants on one or the other allele, so we know which allele came from. And we can see exactly what we know is supposed to be happening here. On the maternal allele, we see a lot of CTCF binding in this region. And then on the paternal allele, we see no CTCF binding and we see mostly CTCF. So this is a way of looking at haplotypes. How are we doing on time? 5 minutes. Perfect. A little more. So the last thing that was the reason why, one of the reasons that motivated this project was we can look at DNA interactions with the centromere. So the centromere, not one of the most glamorous areas of the genome. You know, most of the genome, not most of the genome, much of the genome has these coding regions, which code genes, proteins, and so forth. But the centromere is a really critical locus of every chromosome, and it really provides a docking site of the kinetochore complex, of assembly of the kinetochore complex, which is a complex of proteins that controls, where the mitotic spindle attaches and controls chromosomal segregation, ensures proper distribution of DNA to daughter cells after cell division. And the centromere is incredibly repetitive. It has a lot of these randomly repeated arrays, and is primarily defined by density of these centromere-specific histones, like SEMP-A, which are these histones that are sort of epigenetically controlled, and along with other centromere protein-specific histones, define where the kinetochore will bind. And understanding sort of the quantitative density of these histones in the centromere will give us the ability to learn more about how the kinetochore forms. I'm simplifying a little bit. The challenge, however, is that up until recently, we didn't really know the sequence structure of the centromere, much less the binding pattern of proteins or histones. But recently, the telomere-to-telomere consortium used long-read sequencing to fill out the last remaining 10% of the human genome that was previously sort of missing because it was highly repetitive. By using nanopore sequencing and PAC biosequencing, they sort of filled in these gaps. And actually, Nick, who I was telling you about, well, on the weekends, he was associated with the T2T consortium and was helping them map the centromere. And so now we have this really high-resolution sequence map of the sequence architecture of the centromere, which is kind of like having a high-resolution microscope to look in and see the unique variants and the genetic variants that might drive some of the epigenetic signaling that creates the centromere, the functional part of the centromere. So while we have this really powerful reference, we still need a long-read sequencing tool to map protein binding or histone modification in that region. So there's a lot of data on this slide, but basically it's a summary of our analysis of centromere protein A, SEMP-A, that nucleosome is one of the defines the functional core of the centromere. And so a couple of things. So the centromere is highly repetitive. It's very hard to align to. The telomere-to-telomere consortium recently used nanopore sequencing to identify CpG methylation across the centromere and annotated this thing called the centromere dip region. So this is a region in their data where there's a deficiency of CpG methylation that overlaps with where we think there's an increase of SEMP-A nucleosomes. This is some, you can use short read sequencing to get some gross analysis of enrichment of SEMP-A, and so we know that SEMP-A should align in the centromere dip region, but the actual molecular density on a given molecule of these nucleosomes is hard to measure with short reads. And so we targeted SEMP-A to female-seq, and we showed enrichment of methyl A that aligned with the centromere dip region. We also could see the dip in cytosine methylation. And importantly, there are some genetic markers in this repetitive region that you can align short reads to, but there's these deserts of 10 kb long that are just exactly the same and are very hard to align short reads to. And we're able to get really good coverage across this hard to align to region. And finally, we can use this data to ask, on any given molecule, what's the density? What's the number of nucleosomes, of SEMP-A nucleosomes per unit base in the centromere dip region compared to outside? And we estimated about a quarter of nucleosomes or SEMP-A nucleosomes inside this region, whereas outside are about 5%. So that was sort of not really the end of the story, but the beginning of the potential of quantifying nucleosome occupancy in these really competitive regions. So one of the things that I'm really focused on, I'm not a centromere person, I'm really focused on making this technology better and useful to people that are interested in it. And so we made a really comprehensive protocol.io page. Annie's working on a computational pipeline for people that are not familiar with analyzing nanopore data, and we hope to put out a protocols paper soon. But if you're interested in using this technique, we make a bunch of this PA high of 5, we'd be happy to send it to you and work with people to use it, because that's what I'm interested in, is getting this tool to work better and better and characterize the resolution and the sensitivity. So I'll stop there. This is my lab. We're pretty much exclusively grad students. I'm interviewing a postdoc right now, but we've been grad students for the past 5 years and we're very successful. 3 people have graduated. Nick graduated last year, and now he's working as a Hannah Gray Fellow at Berkeley. Zoe graduated last year. She did that thymus work, and now she's doing a postdoc at MIT. And Anushka, who didn't graduate when we took this picture, but just graduated in December, is now working for 10x. And after 5 years of working closely with Anushka about single cell RNA sequencing and exchanging ideas, now she can't tell me anything that she works on. She works for a private company. I feel like it's a little ironic. Very happy. And then I'd like to thank my collaborators and my funding sources. I'm happy to take any questions. Thank you very much.
Thank you so much, Professor. Thank you so much, Professor Stritz, for this fantastic presentation. Please follow us on social media to receive the latest updates. You can see our social media live. And okay, we have
Anybody from the audience, and I guess we have some online as well, but you can.
There are no online questions.
I'm sure our live audience has questions, so we'll do our best to answer as many as possible. So, we have any online questions?
There are no online. Yeah.
Excellent. Live. I like it. Please.
Why don't you try talking into this?
Okay.
And hopefully it works.
So I thought the total VI method, that was really cool. So I was wondering what's the benefit of that over TSNE or UMAP?
So we use UMAP at DENAS to reduce dimensionality from 20 down to 2 for visualization. So we use some of the, you can use TSNE or UMAP. I think that some of the benefits for total VI is more about how we define the latent space, how we define, so one of the important things, I think maybe the biggest benefit is that, I'm just gonna pull up the slide here. If you look at RNA, you have this sparsity, you have a lot of zeros, but we're not. Just trying to show the stuff.
Oh, I can do that.
Now you're missing it. So you have a lot of zeros. Most of the cells, many of the cells have zeros. The protein measurement has a completely different noise profile. Yeah, it has, instead of a bunch of zeros and a bunch of positive, you have your zeros, there's a finite signal, there's a background. And with flow, typically what you do is you adjust your PMT and you sort of just normalize this out. But with total VI, we quantify the background signal, and we can ask not only what the likely value of that protein is, what's the likelihood that it's foreground or background, because you might have some cells that are negative for the mark, but have positive, enough background signal to make it harder to distinguish. And this allows us to kind of more accurately quantify the abundance on the protein side. If you were to just take the RNA, I'm seeing this quickly, maybe it's not good to do. If you were to just take the protein and the, yeah, I do. If you were to just take the protein and the RNA matrix and concatenate them and then do PCA or TSNE directly without, accounting for the distribution, then the cells would separate out, much more by the batch effects. And so this is a way of dealing with those categorical covariates and those continuous covariates like cell cycles before doing the TSNE. In theory, you can make arguments about whether or not this works or not. We try to prove that.
Yes, please.
So when you were looking at the T cell differentiation and you chose to use the mice modified to only allow for one differentiation pathway,
Yeah.
how could you be sure that having that modification didn't, alter to something else downstream? Like, how can you be sure that that's a direct comparison?
Yeah. Yeah, I mean, this is a good question. I mean, these mice models have been used in a lot of studies about T cell development. And we can do differential expression between them and ask like what's different. The argument that we're trying to make here is by selecting out all the cells that we didn't care about, by filtering those out, and by identifying the different mice as different batches and trying to remove that variation using total VI, and therefore showing that the double positive cells do align in this space with the cells from the wild type. And we're hoping that we've removed the stuff that you're talking about, and we're looking at the trajectory. Now, that could be not true, so that's why we had to go back and ask if the hypothesis that we generated from the lineage mice worked in the wild type culture. And so that's, in fact, that's one of the questions that the reviewer asked. So now we're going through trying to prove that this list of targets and these hypotheses that we generated are valid in the real world. And so that's what's cool about having that new panel of surface antibodies that we can use to test some of those questions that we identified here.
So that was an assumption that you made to begin with, and if
Yeah.
that had turned out to not be
I mean, there are
true,
other things in the literature
Found out
that
later.
gave us confidence that these should be good models as well. But you're right, then we had to go back and ask, are we seeing things that are real in the wild type system?
Thank you.
Well, great talk. I guess going back actually to this figure. So I was interested in thinking about intermediate or stability of intermediate states, right? So I think you showed nicely that when you block CD4, those, CD8 emitted, try out, find a state where they're trying to go through CD4. I guess I was wondering when you're looking at the pseudotime, is there might be more of a comparison between your trajectory wild type overlaid with the CD4, right, with the CD4 cut trajectory, but do you see any functional difference between going back to double positivity, or is it slightly?
You mean like before positive selection, like upstream of it?
So you go from double positive, try out for CD4, then it looked like, at least on the in silico flow, you kind of go back to double positive.
Oh, yeah.
Does it seem like it's exactly the same double positive?
No, and in fact, it's not at all exactly the same. On this two-dimensional plot where you just have CD48, it's the same. But, and there's been other characterizations before this where these cells that are coming back too are distinct. It's just that with CD4 and with the panel that we usually use, it can be hard to identify those. And now we can actually grab those with the pseudotime and do differential expression and ask what are some of the unique transcriptional changes. So these cells actually have already committed to CD8 and they're already expressing like some of the master regulators. But it's harder to see that in some of the flax plots than before. And so we kind of did a lot of this to validate things that people already see.
Well, I guess following up on that, then, was there anything that jumped out besides NFAT that might be that extra little bit of signal?
Yeah, so this is sort of the next step. But all of these, there's a handful of the TCR signals. You see EGR1, you see CD69. A lot of the TCR transcription factors are enriched in the top of this ranking that Zoe did. But some of them are very much involved in both pathways. And one of the things that's really interesting is you see this upregulation of TCR signals in the CD4 branch early, but then after commitment, TCR signaling is also really important for specification CD8. So if you perturb the certain TCR pathways, you're going to block, commitment to either of these. And so we're trying to figure out the timing of that. And one of the ways that, in response to another reviewer, that we're addressing this is by doing some EDU labeling at certain time points and then seeing if we can pull out those cells and see which ones are going which way. That's a hard question. That's the hope is that this data set will provide some transcriptional backbone to the well-known surface protein data to allow us to look a little bit deeper in high resolution. I just opened this up to make sure if there was new questions in the chat.
Okay. That's all the time we
Okay.
have for today. I'd like to thank Professor Streets once again, and thank you all for attending. I look forward to seeing you next semester for your week.